Hello and welcome to part 3 of our notes on muscles. In this one, we're going to take a look at the actual muscle contraction. What's the physiology of causing the muscle to shorten? Okay, so we're talking about the nerves, electrical signals, all that stuff in this one. All right, so how do they contract? So we've got down here in this image, we got the thin filaments, the blue actin, and the red, the thick myosin. And in its relaxed state, the actin and myosin don't overlap a ton, right? They're kind of like if this was an actin and this was a myosin, they're overlapped a little bit. Okay. In the relaxed state, when they get signal contract, what happens is they overlap more, and it shortens up the sarcomere. Like see my arms, the elbows are now closer together. That's contracted. This is relaxed, contracted, relaxed. It's the overlapping of the actin and myosin filaments that causes that the overall muscle shortening. Okay. So here we can see it in this image. Going to go, yep. So when it gets stimulated by the nervous system to contract, the myosin heads here in red are going to bind to the actin in blue and start pulling those actins towards the center of each sarcomere. And it's going to do that several times, not just one movement. Like if this is the myosin, it's not just one movement. It moves, releases, moves, releases, moves, releases, and pulls it in. And that shortens up each sarcomere. Okay, and you see that here. Here's the Z line when it's relaxed, and here's the Z line when it's contracted. And you can see it's significantly shorter. Now, it's not like from here to here, but that little bit of shortening is enough. Okay, so you can see it shortens it up, brings it in. There's just another view of one. There's the thick myosin. Here's the thin actin. And we can see what happens when we have, there's one sarcomere. Now let's start putting them together in series to get the whole myofibril here. And now we can see the whole myofibril relaxed and contracted. We can see how much shorter it gets by doing that. Now, so each muscle doesn't contract a ton. Like if I put my thumb up here at the insertion and my pinky as close to the origin as I can, or the origin of the bicep and the insertion of the bicep down here, when I contract the bicep, you can see that my fingers are not coming together all that much. That little bit of movement of the muscle is enough because of the levers we've set up in the skeletal system to cause a large movement by the time you get to the end of my hand. So muscles don't have to contract a ton in order to get a pretty big movement in the body, at least for skeletal muscles, because they are connected to the skeleton with the joints and levers, so we get that mechanical advantage going on. All right, so let's break it down. How do we get that shortening of the sarcomeres, and thus the myofibril, and thus the whole muscle, to happen? Kind of break it down to 3 steps. One, it's got to be stimulated by a nerve ending or an external electrical source, like if you go and you get those electric shocks at the physical therapist to get your muscles to twitch. That'll do it too, but naturally, a nerve ending will have to stimulate it. 2, the muscle cell itself has to propagate an electric current, otherwise known as an action potential, along the sarcolemma, along the cell membrane surface, and then down into the muscle cell so it can get to the parts of the muscle that are going to contract. Then you're going to have a rise in calcium levels inside each muscle cell, and that rise in calcium levels is going to trigger the actual contraction of the actin and myosin filaments to shorten up the sarcomere. So how do we get a nerve stimulus of this whole action? So skeletal muscles are stimulated by motor neurons of the somatic nervous system. So these are neurons that are connected to muscle. That's why I call them motor neurons. And we'll learn more about the somatic nervous system and all that when we get to the nervous system right now, in the future. Right now, just remember it's the somatic nervous system and motor neurons. And then we'll be able to bring that back together when we get more detail on the nervous system. So the axons of motor neurons. So here is a neuron. We have the cell body. These are dendrites. This is where signal comes in. And then the axon, usually one long branching or one long nerve fiber, this is what we think of as a nerve fiber, comes out, and it will branch at the end as it enters the muscle. Each axonal branch forms a neuromuscular junction with a muscle fiber. So neuromuscular means a junction between the nerves and the muscle. And each one of these endpoints connects to a different muscle cell or muscle fiber. So that'll stimulate one muscle cell to go, that'll stimulate another muscle cell to go, that'll stimulate another muscle cell to go. So when this one nerve fires, it's going to cause 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 muscle cells to contract. So one nerve, in this case, will control 12 muscle cells. Now we can have nerves that control way more or a little bit less than that. And we'll get into that a little bit later. We talk about how we control the strength of contraction, but that's the basic anatomy setup of it. Here we can see a really cool microscope image of branching axon terminals coming in. And then here's a neuromuscular junction where here's a skeletal muscle, right? Long, striated, multinucleate. And there's a neuromuscular junction where it attaches to that particular muscle cell. So we can see that one nerve is attaching to all these different muscle cells. Here's another view of it. We got the nerve fiber coming in, branching at the end, and all these different muscle fibers there. All right, so what happens at this neuromuscular junction? So the axonal endings, which when they get to the end, they're going to have small membranous sacs, not snacks, sacs called synaptic vesicles. These are basically part of the Golgi apparatus producing these in the cell, if you remember those from before, when they had no membrane system. These synaptic vesicles will contain the neurotransmitter known as acetylcholine, which will abbreviate ACH. That neurotransmitter, it means it's just a molecule that's able to transmit a signal from one nerve to another nerve, or from a nerve to whatever it's going to stimulate, in this case, a muscle. Okay, it's a molecule that can carry that message, so to speak. It's a signaling molecule. Okay. Now, the nerve attaches to the muscle at something called the motor end plate. That's the attachment point. And I use the attachment point loosely. That's a specific part of the sarcolemma that contains acetylcholine receptors. So it's got little proteins on its surface of the cell right there that are designed specifically to be the right shape to grab onto. Let's say this is acetylcholine. Acetylcholine can come in and will bind to that receptor and tell that muscle that it's getting stimulated to contract. Though the axon ending and the motor end plate are very, very close, they actually don't touch. They're separated by something called the synaptic cleft. The synapse is where one nerve attaches to another nerve, or in this case, where a nerve attaches to a muscle. That space between them, which is where the acetylcholine is going to drift through, is called the synaptic cleft. They don't touch. So we can see here we got one nerve coming down. A little misleading here. It looks like this one nerve is splitting and it's attaching to this muscle in 3 points. They really should have this attaching to different muscle cells. Anyway, there's the axon terminal. This would be the motor end plate. So let's zoom in on that. So here we can see the axon terminal coming down. There's the axon terminal. And this is the motor end plate down here. You can see the sarcomere and stuff down on the bottom. So let's figure out what's going to happen now. So we got a signal comes down the nerve, gets to this point. What happens next? When a nerve impulse reaches the end of the axon at the neuromuscular junction, we got a series of steps. And I'll tell you right now, guys, this is what your essay is going to be on in this unit. This stuff right here. So we've got voltage-regulated calcium channels. Open and allow calcium to enter the axon. So what do I mean by voltage-regulated calcium channels? So we talked about this way back when we talked about diffusion and osmosis, and when we talked about cell structures like the cell membrane. There are proteins. Now, the cell membrane is supposed to regulate what goes in and out of it, right? Some things can pass right through the cell membrane, the phospholipids. Others need protein channels that are just always open, that let things just move from high to low concentrations under their own power. And some things need some help. In this case, calcium, okay, definitely it's an ion, so it can't pass right through this, the lipids. It needs a channel to go through, but this channel has the ability to open and close to keep calcium from getting into the cell, or to let calcium get into the cell. It's going to go down its concentration gradient. It's got a higher amount of calcium outside the cell. And that's controlled by the electrical signal, the voltage coming down that nerve. The voltage comes down the nerve, hits this protein, that causes the protein to change shape. Calcium now has a clear shot to get into the cell. That's what we mean by voltage-regulated calcium channels. They're regulated by a voltage. Electricity opens them up. And we're going to see that a couple more times here in this muscle section and a whole bunch when we get to nerves. So once that calcium gets into the axon terminal, it's going to cause axonal vesicles full of acetylcholine to fuse with the axonal membrane. So these are the, so calcium gets in. You see it right there through these voltage-regulated channels. And these vesicles right here are full of acetylcholine, and they're going to bind to the membrane of the axon terminal and release their acetylcholine into the synaptic cleft right here. We see it over here. Here's the acetylcholine getting released into this cleft. So, like I said, it's going to let that acetylcholine get into the cleft via exocytosis. It's a vesicle opening up and pushing it all out at once. That acetylcholine will diffuse across the synaptic cleft. It's not a very big distance, but there is a distance there, so it does cause a ever so slight delay to the acetylcholine receptors of the sarcolemma. When the acetylcholine binds to those receptors, it's going to initiate an action potential in the muscle, which we'll talk about in a minute, and which is basically it'll cause the muscle to generate an electrical signal. The simple way to put it. Now, you don't want that acetylcholine to stay bound to that receptor. Otherwise, your muscle is just going to keep trying to generate electrical signals, and you're going to get stuck like this. Big old cramp, or one version of how a cramp happens. It'll get stuck there, and you won't be able to release it until you run out of juice, okay? So that acetylcholine, once it attaches, you have to destroy it and break it down, and there's an enzyme in there which will do that in that cleft, which will break it down and get it out of the receptor to clear it so it's ready for the next signal. So we got calcium came in, causes synaptic vesicles here to release acetylcholine. Acetylcholine comes across, binds to these receptors. Now, these are kind of like those calcium channels, right? These receptors here, these acetylcholine receptors, when the acetylcholine attaches to them, they'll cause these receptors to change shape and open up and allow sodium and potassium to flow through. So potassium ions to flow through here. So this, instead of being a voltage-regulated gate, this is a molecular regulated gate, a neurotransmitter-regulated gate. That's how most neurotransmitters work. So you've got this receptor for acetylcholine. Acetylcholine binds, causes this acetylcholine receptor to open up the gate that's attached to it. I can't do that with just my hands, but I'll open that gate. And now, as long as you have a concentration difference of sodium and potassium on either side of that cell membrane, they're going to start zipping through, okay? Going down the concentration gradients, you can see sodium is going to be going in, potassium is going to be going out, and these are ions. So those ions are moving rapidly. And what is electricity, guys? Well, the electricity is powering your screen right now on your computer that you're watching this on. That electricity is moving electrons, right? Electricity isn't just moving electrons, it's moving charged particles. The electricity we use in our buildings and stuff, those charged particles are electrons. But in biological systems, the electricity is generated by moving ions, charged particles. So sodium, potassium, if you get enough of them moving quickly all at once, you will generate an electrical signal, a voltage. Okay, so that's kind of the heart of this. I'll do a drawing and stick it in the video here in a little bit to kind of help illustrate that a little bit. So we've got this electrical signal. If enough acetylcholine attaches to the receptor, enough. If just a little bit dribbles across and attaches, nothing's going to happen. It's just not going to be a strong enough signal to get things to move. It'll cause that sarcolemma, the cell membrane of the muscle, to become temporarily permeable to sodium ions. So sodium will be able to zip through that membrane and go all the way down the length of that membrane and generate that electrical signal or that action potential. And once you reach that threshold, okay, the current will flow through the entire cell and it will contract. And like I said, I'm going to do a drawing and insert that in here either before this or after this. So came in here, if we get enough of this acetylcholine binding to enough of these receptors, we'll get enough sodium and potassium zipping through to generate a strong enough electrical signal. If it's strong enough, it'll cause the rest of the cell membrane to become permeable to sodium, and you'll send an electrical signal down. Okay. That action potential is going to travel down the cell membrane, then go down the T-tubules. So it's going to travel down the cell membrane, down these T-tubules, these yellow things, and into the sarcoplasmic reticulum. Okay. The sarcoplasmic reticulum, that is storing calcium ions, and it's able to release them or reabsorb them. When it gets this signal, voltage-regulated calcium channels are going to open up again, and they're going to let calcium leak out of the sarcoplasmic reticulum and into the myofibrils, to the sarcomeres. Okay? And that calcium is going to trigger the next step in the process. So that calcium will cause the actin filaments, the thin filaments, to change shape. So if this is the actin down here, right? It's this shape, and the myosin can't attach to it. It's like slick. When the calcium gets in there, it'll cause the actin to change shape, and now the myosin is able to bind to it and start doing its contraction. It's shortening its stroke there. We'll form what's called a cross bridge. The myosin actin will bridge across the gap, and we will start this whole process. Okay. And that's called the working or the power stroke. The myosin head pivots and pulls the actin filament towards the middle, towards the M line, right? And it's going to keep doing that. Then it's going to detach. And what causes the detachment is actually ATP, that energy storage molecule we make during cell respiration. ATP is going to attach. That'll cause it to release. ATP will expend its energy, which will cock the myosin head, and it's ready for the next stroke. It'll attach, pull. We'll use another ATP molecule to detach it and cock it. Then it'll attach and pull. And it'll keep doing that as long as it's getting the signal, the electrical signal from the nerve, which is then keeping calcium being released into the myofibril, which keeps the actin changed so the myosin can keep attaching. It'll keep doing that either until it runs out of ATP or the calcium goes away. It's reabsorbed. So let's take a minute and watch this video here. This video will show us how this all happens in the cell.
Contraction of skeletal muscle tissue occurs when actin and myosin myofilaments slide past one another, causing the sarcomere to shorten. It is important to note that the individual myofilaments do not change length themselves, but instead overlap each other to shorten the sarcomere. Moving in for a closer look, we can see the details of how this occurs. The molecules you see here are myosin, which at rest has both adenine diphosphate and phosphate molecules attached at the individual myosin heads, actin, tropomyosin, and troponin. When we decide to move, calcium is released from the sarcoplasmic reticulum. These molecules bind with the troponin molecules. This causes the tropomyosin molecules to move, revealing myosin attachment sites on the actin myofilaments. The single phosphate attached to the myosin heads gets released as the heads move to connect with the attachment sites on the actin. The resulting formation is called a cross bridge. The remaining adenine diphosphate molecule is then expended in what is called a power stroke, when the myosin heads pull the actin inward. Once the adenine diphosphate attachment is spent, adenine triphosphate molecules attach themselves to the myosin heads. These molecules trigger the release of the myosin heads from the actin attachment sites and are immediately broken down into adenine diphosphate and a single phosphate as the heads move back into their resting position. This is called the recovery stroke. If calcium is still present, the cycle will repeat again, and the sarcomere will continue to shorten until the calcium ions are transported back into the sarcoplasmic reticulum and the muscle relaxes.
So we just watched that video, guys. Just kind of put it into perspective with this thing here. The actin and myosin create a cross bridge. They connect. The myosin moves and pulls the actin towards the middle to shorten the muscle up. Okay, the ADP gets released. An ATP comes in and causes the cross bridge to get broken. The ATP gets used. You break that last phosphorus off, and that recocks the head, so it's ready to go. It's kind of like a coiled spring, and we start the whole process over again. So just to summarize, electrical signal comes down the axon, down one of the axonal branches to the neuromuscular junction. We're in the axon terminal. That signal coming down causes voltage-regulated calcium channels to open. Calcium rushes in. Synaptic vesicles full of acetylcholine, because of the calcium, will be able to fuse to the cell membrane here of the nerve, release their acetylcholine. Acetylcholine will diffuse across the synaptic cleft and attach to acetylcholine receptors. Those will then, the acetylcholine attaching to the receptors is kind of like a lock in a door. It'll open up the door, which will allow sodium potassium to zip through down their concentration gradients, generating electrical signal. If you got enough electrical signal, you reach that threshold of electrical signal you need to transmit the signal. You generate an action potential. That will cause a chain reaction all the way down the sarcolemma, the cell membrane of the muscle. So you're generally sending electrical signal down the surface of the muscle. That'll go down the T-tubules to the sarcoplasmic reticulum, releasing calcium there inside the cell. Calcium causes the actin to change shape. Myosin heads can now bind, forming a cross bridge. Once they bind, they bring the actin towards the middle. An ATP molecule comes in, attaches to the myosin, causes it to release. Get rid of that phosphorus. Okay, so release that energy. It'll cock the myosin head, and we'll keep doing this until we're no longer getting an electrical signal, and that calcium gets reabsorbed. Once the calcium gets reabsorbed, the actin changes shape. Myosin can no longer grab, and everything slides back out because of those elastic fibers on the end of the sarcomere. And that is a muscle contraction.
Hello, folks. I'm switching over to a whiteboard presentation of the sequences of events in a muscle contraction from when stuff comes in on the nerve until when it's all done. So what we've got here, folks, is we have a nerve over here, right? There's the axon terminal at the end of the nerve. Here we've got the motor end plate, okay, where the nerve and the muscle come together, but they don't quite touch, you can see. This would be the sarcolemma, or the surface of the cell muscle fiber, the cell. And down in here is all inside the cell. Then right here, we've got the T-tubule, and then a really bad drawing of the sarcoplasmic reticulum. And all through here would be myofibrils and sarcomeres and all that kind of stuff going on. Okay. So the first thing that we're going to take a look at is the electrical signal, for lack of a better term at this point. We'll get into that a little bit more. Comes down the axon to the axon terminal, and there are protein channels in here designed to let calcium ions, which I'm going to do with these little black spots, going to be calcium ions. That protein channel is not always open, okay? It's usually closed, keeping all this calcium outside of the nerve. And when the nerve is resting, there's pumps that are going to be pumping that calcium out of the nerve furiously to build up a huge concentration difference, kind of like water behind a dam, just pushing against the dam trying to get in. Remember, diffusion, right? High to low concentrations. Well, this is a voltage regulated calcium channel that I drew here. And when the voltage gets to the end of it, it's going to cause this thing to open up. I don't know how to draw it opening, but all these calcium ions are going to rush into the end of the axon terminal. Okay. Now, when that happens, what we're going to see is inside there are a bunch of axonal vesicles, put together by the Golgi apparatus, right? This is the endomembrane transport system all over again. And inside there, all these little blue dots are something called a neurotransmitter. And this particular one is acetylcholine. That's going to be those little blue dots, okay? They're inside here. All right. And when the calcium comes into the end of the nerve, that causes these to migrate to the end of the axon and release their acetylcholine via exocytosis into the synaptic cleft, into that gap there. All right. As you can see, there is no direct contact between, no direct contact between the nerve and the muscle. There's a gap here. So that acetylcholine has to diffuse across the gap to get down to the muscle. Okay, let's go back to drawing here. And we have the similar gates down here at the motor end plate that are keyed to acetylcholine, literally keyed. Acetylcholine is just the right shape. When acetylcholine diffuses across and attaches, that's going to cause this to open up. And so let's have these all open up. It's like an open door here, okay? They're all going to open up and they're going to be open. Now, they're not opening to let acetylcholine through. Acetylcholine is just the signal for them to open. It's not a voltage regulated channel. It's carrier mediated channel. So they're neurotransmitter mediated channel. So that acetylcholine is attaching and opening those up. So that begs the question, what is trying to get through? Well, what we're going to do next is I'm going to portray sodium ions with little red dots, and I'll do some potassium. So I'm not going to focus on that because it's too much back and forth, with green dots. Okay. Now, when the muscle cell is at rest, there are protein pumps, sodium potassium pumps. I mentioned this when we talked about cell transport earlier in the year, is a very important one. And those pumps are putting a whole lot of potassium inside the muscle cell and a whole lot of sodium outside the muscle cell, all along through this motor end plate area. And I'll just draw a bunch in here. All the way down the sarcolemma, the surface of that cell. Okay, so we've got a huge concentration gradient difference again. Lots of sodium outside and lots of potassium, didn't switch the color, on the inside. So when these acetylcholine neurotransmitters in blue open up those gates, now all of a sudden sodium can rush through and into the cell. These are sodium channels now. It's rushing through. Okay, and like we said before, what is electricity? If it is not the movement of charged particles, and sodium has a plus, it has a charge. It has electrical charge. Now, it's not as small as the electrons, that we got zipping through the wires in our rooms helping us light ourselves. Those are charged particles on the subatomic scale. But when we're talking about biologically produced electricity, we're looking at ion movement. So now we got lots of ions rushing through here very quickly because they're going down the concentration gradient, big difference, lots of speed, like we talked about earlier, and that's going to generate a voltage. Again, and if you remember, we saw that up here with calcium channels. If we have enough voltage, we can open up voltage regulated. Now, these ones I drew down here in blue, those are regulated by acetylcholine, but up here on the sarcolemma, these are voltage regulated sodium channels. Now, just a little bit of voltage is not going to be enough to open these up, okay? It's like you got a garage door opener with a very weak battery. It's probably not going to get it to open, right? You need to reach a certain amount of voltage called threshold. You have to have enough juice to get this thing to open. Now, once you get that thing to open, now sodium rushes in really fast and generates more voltage. If you can get this first one to trip, okay, guys, if you can get that first voltage regulated channel to go, it's always going to let enough sodium through to open the next one. And that's going to go through, which is always going to create enough energy to open the next one. More sodium comes through. The next one, more sodium comes through. This is called an action potential. Okay, potential for voltage, action, because you got enough voltage or potential to make something happen. Okay, and this is going to happen all the way down the sarcolemma, like dominoes falling. If that first one falls, they all fall. All the way down here, all the way down the T-tubules to the sarcoplasmic reticulum. And now we've got that signal all the way down into the sarcoplasmic reticulum. Now we come back to calcium ions in here. Okay, the sarcoplasmic reticulum has voltage regulated calcium channels again. And when they get that voltage, they let the calcium out of the sarcoplasmic reticulum and let it get at the sarcomeres, at those actin filaments, those thin filaments. And that when that calcium gets to those thin filaments, it's going to allow that actin to change shape. So let me just draw an actin. Let's just pretend this is actin, okay? And let's make this myosin. Okay, myosin in here, and it's got its little head on it. Normally that myosin head can't bind to the actin. It can't attach. But if calcium binds to the actin, it's going to allow that actin to change shape, and it's going to allow that myosin head to attach and form a cross bridge. And then it can start moving and start this whole process going. Okay, that calcium is going to allow that connection and the cross bridge formation, and you're going to start the contraction. Now, when we can no longer let calcium out, when we stop getting all the signal coming down, calcium gets reabsorbed back in the sarcoplasmic reticulum, and we can no longer form that cross bridge, and we're done. Because the way these nerves work, guys, your nervous system up here with the nerve, your brain either sends a signal or it doesn't send a signal. There's no like information in there other than the fact that it's on or off. It's kind of binary. So as long as we keep sending signals down this nerve, we keep releasing acetylcholine, we keep sodium rushing through the channels, we keep sending that signal down, and releasing that calcium, we're going to keep that muscle contracting. But the second we stop sending a signal, no more acetylcholine, it's getting destroyed down here so it doesn't get stuck and keep these open. No more acetylcholine, these close, no more electrical signal. These voltage regulated calcium channels that are letting the calcium out close, and we just start pumping the calcium back in. And then this can no longer happen. We can't form the cross bridge, and we can't continue the contraction. Your muscle relaxes. And that, my friends, is how we cause a muscle to contract.
There you go, guys. We'll talk to you later. Bye.